Thanks, Dennis. Everybody hear me okay? Raise your hand if you can't hear me. I don't know why people say that. It never works. Well, thanks very much for coming out this evening, and thanks again to Dennis and Harbor Branch for having us. What we're going to show you tonight is a perfect example of applied management, where we take information from the laboratory and apply it to the field. And we've done this collaboration for a good reason. I work for a state agency, Fish and Wildlife Commission. We handle all the fishing and boating laws of Florida. And Josh, of course, works here at the university system, a research system. And so it's a good partnership for us to take research on a coral reef and to try to use that to educate water managers about what's best for our state resources, in this case, corals and fishes. So the story on corals worldwide is unfortunately a rather sad story. It's estimated within the last 20 years that we've lost anywhere between 1/3 and 1/2 of all the corals on Earth. And that's mostly a result of climate change, and we can point to specific events like El Nino events, very hot summers, for example, around the tropics where we've lost a lot of corals. And when we say that corals are photosynthetic organisms, that isn't exactly the truth. It's in fact the tenants that live inside their tissues that are doing the photosynthesizing. And it's these tenants that are called zooxanthellae. They're basically just a specialized algae that live in the tissue. So this is a commensal relationship where both species are benefiting from this relationship. So the zooxanthellae are providing food to the coral, and the coral is providing them a place to live, as you might say. And corals live at the very threshold of their survivorship, in that they live in tropics and subtropics where the water temperatures hover somewhere around 80 degrees or more Fahrenheit. But for most coral species, if it hits 90 degrees, they show signs of stress and often will die. And so it's this concept of coral resistance and resiliency that are the main buzzwords in the coral research world right now. So as you see on this slide, for example, on the left-hand side of the slide, are several colonies of the same species of coral, and we wonder why this particular coral responded negatively to a stress, whereas the other corals in the same species seem to be doing just fine. So here's an example of resistance happening, where somehow these corals are able to resist whatever stress is happening, and this one is not able to. And going back to that last slide that I showed you, when the coral shows signs of stress, unfortunately, they don't show any visible signs until they're dead or mostly dead. And that's what's happening with this particular coral here. It's doing something that we call bleaching. So in this case, it's evicted those tenant zooxanthellae as a result of this stress, whereas the others in the picture did not. And then secondarily is this question of resiliency. We wonder, why is it that some corals can, as this one, which is a picture taken from St. Lucie Reef, the reef we're going to introduce you to tonight. Why is it that this coral was able to only partially bleach and then respond to such that it did not lose any tissue? So what can happen in this case is the zooxanthellae were evicted from the tissues. There were still some left in the tissues. They were able to reproduce when water conditions got better. The coral was also possibly able to take up zooxanthellae from the water column. So this one completely responded. It was resilient to the stress. And when we went back months later, this coral looked like it used to. And these were no longer bleached. They were the color they were supposed to be. So when we think about corals and coral reefs, those are the two main topics that we were discussing. And one of the big questions for us is, are there other indicators that we could look to before the coral gets to this terrible state? So it's this idea of indicators of sublethal effects, effects that are before the coral dies. And what we want to introduce you to tonight is the northernmost coral reef in Florida. And we say that because most of the hard corals that occur on this reef do not occur north of this particular reef. only about 5 or so species occur north of this reef, whereas we have about 25 that occur on this reef. And so for that reason, it's considered the northernmost coral reef in Florida. And for those who aren't familiar, this is the St. Lucie Watershed. So this is our backyard. And of course, probably the most interesting and famous, or infamous, I should say, aspect of this watershed is our connection to Lake Okeechobee. This watershed was not connected to the lake historically. This is a man-made feature that happened in the early 1900s. So we have an example which is rather rare globally, and that is the ability for lake water to move all the way down onto a reef. And I show this as brown water. I should make the distinction. The St. Lucie system is a black water river, as are most rivers in Florida, in that it should be full of tannins from plants that grow within the watershed. So think about coffee or tea as an example. But it shouldn't be chocolate milk brown. And that, unfortunately, is a situation we often have here. So a lot of sediment and excessive nutrients in the water that make it very brown instead of black. But as you can see, the C-44 canal that's attached to the lake is not our only problem with this watershed. In fact, if you look over the last 50 years, you can explain only 1/3 of the volume of the water of the St. Lucie as coming from the lake. The other 2/3 comes from the other canals. And it's entirely possible for us to go for several years without any releases from the lake and still have major problems in the estuary and on the reef. We have performance measures for restoration of the St. Lucie, and they have a lot to do with the salinity range. For example, at Roosevelt Bridge, the US 1 bridge there in the middle of the estuary, we'd like to keep that somewhere between 10 and 20 parts per thousand salinity. Full strength seawater is around 35. We can get out of that range, so we can be less than 10 parts per thousand for many days or weeks without any releases from the lake. And so it's these other canals that are also of concern to us. And by the way, quite a bit of water comes within the watershed of the C-44 canal itself and goes into the St. Lucie. Again, nothing coming from the lake at that point. So the C-23, C-24 canals, and then the North Fork are also major contributors to this basin. And then we can think about all the other tertiary canals associated with our neighborhoods. So quite a bit of volume going into the St. Lucie. And it's the major releases, especially the ones from the lake, that make the news. And so here's an example. We're in the late 1990s, which is our last major release event, where we had lesion fishes. And we know for certain that the lesions on the fishes are related to low salinity, high nutrients, probably a fungus that's affecting the fishes. So lots of problems within the estuary. So here's how we manage the lake. And as you can see, it's very simple, straightforward. So this is a very complex system. That's the point of this slide. And there are numerous agencies involved in this process. The process is primarily driven as a cooperation between Army Corps of Engineers and South Florida Water Management District. But they work with all the other agencies, including ours, to try to make the best decisions possible, given that it's a highly managed system. And there's one word you'll see repeated on this slide a lot, and that is the word forecast. So a lot of this has to do with trying to guess the future. And we all know how weather forecasting goes. And then one of the questions we get about the St. Lucie is, why haven't you come up with a plan and fixed this thing yet? You've been studying it. We don't need any more studies. We need some more action. Well, in fact, the study plan was done in 1998. And this slide is somewhat of a synopsis of that plan. It's referred to as the Indian River Lagoon South Plan. And it's our local neighborhood version of the Everglades restoration, the SERP. So we know what it takes to fix this basin. By the way, the price tag is now $1.5 billion. And basically it's to put in reservoirs and stormwater treatment areas that store the water and cleanse the water. So releasing it when it should be released and releasing it in better water quality than it is currently. And then also doing things like plugging ditches on some large pieces of property like the green one you see there, Alapata Flats. So the plan is in place. Just a few of these features have been done or are currently being installed. But basically, we still have a long way to go in order to achieve what we want within the watershed. So here's how the estuary and the lagoon works. Again, very simple. Makes you wonder why restoration is taking so long. No, these are very complex systems. And this reminds me that we've had a lot of seagrass loss in the north end of the lagoon. We could use this same ecological model for the north end of the lagoon as well. It has all the features there. So when we lose seagrasses as a result of algal blooms, as we did within the last few years, very complex question for us to be able to understand how it happened and how we can keep it from happening in the future. But I mainly want to direct your attention here to the bottom, and that is the fact that everything flows downhill. So it all eventually ends up on the nearshore reef. And for the most part, we understand performance measures for restoring an estuary. We basically know what seagrasses need in terms of light, nutrients, et cetera. We know what oysters need. They need a particular salinity balance. They need a certain type of phytoplankton, certain size range of phytoplankton. But we really don't know much about what reefs need to survive. And so one of the things we're trying to learn here is what makes this reef so unique and resilient, and what does it teach us about reefs in general. So that's one of the main questions we're trying to develop through this is what are the performance measures for restoration of a reef? And this slide I'm going to go through very quickly. This is Brian Lapointe, the guy that some of you hadn't heard of yet. Here's some of the work that he's done. Brian has worked on nutrient input to places like reefs. And the main point of this slide is this. When you compare St. Lucie Reef with all the reefs to the south, unfortunately, it wins in all the wrong categories. It has the most algae, which of course is going to respond to a lot of nutrients, and has the highest levels of things like nitrogen and phosphorus. So that reef is in a lot of trouble. So let's look at the reef specifically. So this is a LiDAR image, laser image, taken from an airplane, to which we can really see a good picture of the bathymetry. And up near the inlet, the north end of the reef, you find a predominance of worm rock that's shown up in the corner here. This is sort of a rare, at least a globally rare organism. So it's a colony of worms that are able to grab sediments from the water column and secrete a glue and create somewhat of a rock formation. Very interesting. So as you can imagine, a lot of sediment coming out of the inlet, so a good place for worm rock. As you reach down to maybe the middle of the reef here, that's where you start picking up some of the larger corals, including brain corals. This one is one we call Diploria gloria. It's 14 feet in diameter, largest brain coral I've ever seen. And we know that this coral is at least 100 years old because we cored it. More information on that in a little bit. And as you go further down the reef, you pick up some of these other interesting species like mountain corals. And it's these two that are of most interest to us, the brain coral and the mountain coral, for the purposes of this particular study. So just let me give you some facts on the reef itself. Over 250 species of fishes, which is very interesting. It's a very diverse community. Like I mentioned earlier, about 24 species of hard corals on this reef. Caribbean-wide, about 75 corals. In the Keys, about 50 or so. So this has about half that, 24. Very few soft corals, probably because of the wave action and water quality issues. A host of invertebrates, including the long-spine sea urchin. There was a massive die-off of this organism throughout the Caribbean in the 1980s, probably as a result of a virus. And for some reason, this reef seems to have been spared. And then, of course, lots of algae for those critters to eat. It is also a state park. Wanted to point that out. There's a commercial fishery happening there. So we're working with the fishermen to clean up nets. But here's one of the main points, the slide I wanted to draw your attention to, and that is the invasion of lionfish is teaching us a lot about larval transport on this reef. Perhaps one of the reasons why there are so few corals on this reef is because so few coral recruits are brought to the reef. We've only captured 30 lionfish on this 5-mile stretch of reef in the last few years, despite perfect habitat for them. So we're learning lots as a result of the invasion. So here's a picture on a good day, before discharges on the reef. And then here's what it looks like. Can look like afterwards. So if you remember, summer of 2004 was a good summer, beautiful weather, but we didn't know it was coming that fall. And that was the hurricanes, 2 of them in a row, if you remember, Francis and Jean. So here it is before those events. You can see the hard bottom there clearly. Now here's what it looks like afterwards. And this was in July of 2005. So this is roughly 5 months of lake releases happening at that point. So amazingly resilient corals are able to survive this. And it was after those release events from the lake, following the hurricanes, that we had, to my knowledge, the one and only bleaching event on the reef. So again, those corals are evicting their zooxanthellae and their tissues and showing signs of stress. This happened in the winter. Bleaching normally happens in the summer. It's a combination of temperature and light, usually. In this case, it looks like it was just a reduced light event because of dark water. Corals are interesting because they also lay down their calcium carbonate skeletons, much like a tree. So if we take a core of those skeletons, we can look at the growth rings. So here's an example of that, of a brain coral in the central part of the reef that we took. And the point of this slide is just to show that rainfall is driving the flow. No surprise there. But the interesting point here is that before 1970, there were just tremendous amounts of flow. It wasn't until 1970 that we really started doing water management. Can we do a better job? Certainly. But that's the point of that one. And as I mentioned, these corals are laying down these bands of high density and low density, which would be dry season and wet season respectively, so that we can learn about their growth. So things that we look at is the amount of calcium, the size of one of those bands, because that's indicative of how much growth has happened in a year, and the density. So in other words, it's a reflection of how many other micronutrients, lots of minerals and metals, can be placed into the skeleton. So if we look at flow versus calcification, it's significant that as flow increases, calcification decreases on this reef. Extension rate, same story. As flow increases, the extension rate decreases. So clearly, these corals are showing signs of stress as a result of the flow. Density, no significant relationship, which is somewhat surprising because usually there's a significant relationship there that the density will increase as flow increases. We monitor the corals as part of a comprehensive program that goes from here all the way to the Keys. And so the point of this slide is simply that St. Lucie Reef has fewer number of species of corals than our neighbors to the south. But in its densest places, it has as much percent cover as some of our places to the south. It's just that the corals are concentrated into specific areas that are just perfect refuges for them. So we look at flow and salinity, and here's just data that we got that's collected at the inlet by the Water Management District. You see a really nice relationship there, that as flow goes up, salinity goes down. No surprise. And at the inlet, it can be as low as 1 part per thousand. Seawater, again, is somewhere around 35. So we can take the St. Lucie all the way to fresh water, top to bottom, all the way to the inlet when we have major releases. And this is flow versus Secchi depth. Secchi is the little disc, black and white disc that we use to test for water visibility or clarity. Same story, as flow increases, visibility decreases greatly. And within the park, here's an example of a pretty moderate flow event, not as high as some of the ones in the past, but fairly high. Within the park, the Secchi depth would be less than 1 meter. So the visibility is less than 1 meter within the park. So it's amazing that these corals can hang on. So here's the reef and the sites that we're studying. North, central, ledge, and south. And then along the dune, for some of our light studies that we're going to talk about here in a minute, we like to use a light sensor to get an idea of cloud cover, because that obviously plays a role. So we consider all the things that are important on the reef. Of course, the amount of sunlight. We're at a pretty high latitude for corals, not the highest. Places like Bermuda and Flower Gardens are even higher latitude. But these corals are already suffering from less light than their friends to the south. A lot of wave action on this reef. It's only 4 feet deep at low tide in places, so wave action plays a big role. Cloud cover, and that's exactly where the storms happen almost on a daily basis. And then one thing I had mentioned before, and that is upwelling. That is deep ocean water that's cold and often nutrient rich, also moves onto the reef, generally in midsummer for a 1 to 2 week period. Another stress on these corals. And if that weren't enough, of course, we add to it this issue of dark water coming out of the inlet. And In the old days, I would have said to you that it's a very simple system that the water comes out of the inlet and just flows to the south. But it's not that simple. Spending time on that reef and doing research, we've realized it's very complex. And wind speed and direction plays a big role. So example, on a southeast wind, that plume would not be coming south at all. It might be going straight out the inlet. And you can get pockets of dark water that end up maybe at the central site, but not at the northern site. So it's not as simple as the north corals are the most affected. It's much more complex than that. And one of the ways that we study the reef is to look at the amount of light. So we have various instruments that measure light. And we use those instruments to calculate light attenuation. So the take home from this is that in every case, the north site has the most light attenuation, in other words, the darkest water. And then when you get into the late summer and fall, when the dark water is happening, a lot of discharge is happening, you get to the point where you get some pretty high levels of light attenuation. And another way that we can display this light data is to look at maybe a couple of consecutive days. So this is in May at the north site. Based on our dunes, we can see it's a pretty nice day, not too much in the way of cloud cover. And the corals are getting about 3,600 lumens per square foot of light, and high tide here being at 1124. But when we move to the fall, same site, but now we're in October, we've got black water and we've got wind. Now we're down to only about 1,000. So big difference that happens as a result of the black water and the amount of light that are getting to these corals. And you might ask me at this point, well, what's the minimum threshold for corals? How much light do they need? Well, we don't know. So it's one of the things we have to continue to study. So if we look at all the sites here together, north, central, and south, and the light, and this was through summer of 2010, there's a couple of interesting stories for us. One is that during April and May, 82,000 cubic feet per second were released. So you can see that it was affecting these sites. Another 39,000 released into June. There's the upwelling that I mentioned happens midsummer. It really dropped the light out. It was dark, flocculent, cold water that came up. And then lastly, these storm events, which start in the fall, the wind starts moving around the dark water, and then it really drops the light out. So probably the toughest time of year then for these corals is this fall period. If they can make it through the fall, they can probably make it through the rest of the year. And one interesting dynamic about this is light on the reef is dependent on the amount of water that's coming in by tide, as well as the amount of water that's coming down from the discharges. So as tide comes in, it's bringing in clear water, but it's also making it deeper, right? A greater volume of water is coming in. And so the take-home slide from this is that for the north and central sites, before the upwelling period, in the dry season, when the water should be clearest, as tide stage goes up, the amount of light goes up. So it's bringing clear water into those sites, even though it's getting deeper, because it's so clear, it's providing more light. The south end, that's not the case. As tide stage goes up, light intensity is going down. It's already clear at the south end most of the time, so as the volume comes up, it's getting deeper, it gets less light. At the end of the summer, when things are getting dark because of that dark water, all the sites behave like the south side. So clear water is trying to push its way in, but all it's doing is moving the dark water around, and you're not getting more light with clear water, unfortunately. So that was a lot of technical slides. Take a deep breath here. Take a seat on the reef. There's a chair that was provided by one of the locals we found out there one day. So let's just think about all of these implications. What is it like to be a coral on this reef and to survive? Obviously, the reduction of light from all of these forces are going to be important. So things like color, turbidity, that is the amount of sediment. Corals have an amazing ability. They are, of course, little animals. Think of them as a colony of anemones that they can push sediment off of them. But there's only so much sediment that they can take. Things like dissolved organic matter. All those things are part of this stew that's reducing light. Also, there's the issue of osmotic shock. One of the things I failed to mention was the temperature can drop 17 degrees in 17 hours, as it did a couple of summers ago. So there's all these issues of shock, both with salinity and temperature. These excess nutrients like nitrogen and phosphorus and dissolved organic carbon, they all affect things like dissolved oxygen. Of course, these are animals, they need oxygen. Sedimentation, of course, is going to increase competition for other organisms. As I showed you earlier, you've got a lot of worm rock at the north end of the reef. Well, the worm rock really enjoys that heavy sediment. That's what it's building the rock from. So there are certain organisms that can outcompete corals under these sorts of conditions. And then lastly is a whole host of things that we really know nothing about on this particular reef especially, and that is bacteria, heavy metals, pesticides, herbicides, all these things that are detrimental to organisms. So I say all that to say this: it would really be nice if we could have some sort of coral stress early warning system. Instead of finding a coral that's bleached and realizing, ooh, something's not right, maybe we should have done something water management-wise. It would be very nice if we could have some other indicators. And so that is a perfect segue into Josh and the work that his lab is doing.
Everybody hear me okay? All right. Make sure the pointer is working for me too. Okay, so Jeff talked a lot about the kind of longer-term data and longer-term studies that we've done on this reef, and most of those that are fairly coarse scale, giving us information about the total flux of water as it comes out of the inlet, or giving us information about the total amount of light available over the course of our sites. And we realized that to really understand what's happening to the corals on a physical level, and more so to get down to the point where we can provide data about the responses of the ecology to a management action, we need to be working on the scale of days to weeks to months rather than years. And there's a couple of important reasons for that. Number one, we want to be capturing these events before they kill the coral. Number two, political cycles don't work on long time scales. They work on short time scales. So if someone makes a decision about a management action, they need data that shows that what they did worked in order to get reelected. Otherwise, they're not going to do it, right? So we started to think about how we could put in different kinds of sensors and different kind of metrics to get that shorter and finer time scale data. And the first thing we wanted to do was really track those pulses of fresh water as they came out of the inlet and all the way down and across this reef. And the best way to do that was to install individual salinity loggers at every single location. Thankfully, I had a technician, Maureen Williams, who spent quite a bit of time doing research on salinity sensors all over the world to try to find an alternative for us. And we ended up sourcing a sensor from New Zealand, these Odyssey sensors. And we deployed those this past fall, and they've worked beautifully so far. So now we finally have good salinity data across the scope of all 4 of our sites, as well as the inlet. And so here you can see our 5 sites again that are color-coded. Those color codes match up to what you're seeing here down below. We're looking from mid-October up until just last week. And here's our salinity going again. Remember, 35 is kind of out just offshore, and 0 would be freshwater. So what you notice here is that we have lots of variability, and when we get dips, it's apparent that most of the sites are being affected equally. So as salinity drops, you'll see it drop at all 4 of our sites. We didn't get the salinity monitor installed at the inlet until a little bit later, starting right here. Anyone know what happened on January 9th? Did your front yard flood? Ours didn't, but our neighbors did. Indian River Drive collapsed, school districts closed, people were stranded all over the place, cars were left, right? We had about, on the order of 10 to 20 inches of rain down into South St. Lucie and Martin County. So we know that our salinity data is picking up potential responses. And we know that there's 2 potential factors associated there, both rainfall and discharges. So we wanted to look at both of them. So if we compare the salinity drops to rainfall, we get some pretty good concordance in that as a rainfall event happens, we tend to see a dip in salinity. But there are some dips in that graph, say like around mid-December, for which there is no associated rainfall. And so that begs the question, is there some kind of discharge event going on that may be driving the system as well? So we want to see if freshwater discharge matches up to any of these drops in salinity that we see. And we've used 5 different locks and the discharge rates from those locks in our overall watershed system. So most of us are pretty familiar with this one. This is S80. This is the one that tends to make the news in the picture that was at the bottom left of the slide with the fish legions. But there's also a lock up here at Lake Okeechobee and additional locks on control structures in C23, C24, and 10 Mile Creek. So if we look at the flow data from these locks in comparison to our salinity data, there's a couple interesting things. First is that in late fall, even as the rainy season starts to taper, we're continuing to have discharges out of Lake Okeechobee. That's the positive red here. And then when we see these bumps related to rain events, we tend to open up C24 and C23 and maybe 10 Mile Creek at times too, and we get some flow coming from the northern fork of the St. Lucie River. One of the interesting things about the big rain event in January, just a few weeks ago, was that all 3 of those northern locks were open, but S80, down here near Stewart, was actually closed, totally flat. All of the water that was draining into this basin was actually directed backwards into Lake Okeechobee. That's why you see that big dip of a red line here on the bottom. So we're not locked into this mindset of all of the water that's in this basin is always going to run down into St. Lucie. The system's constructed to provide flexibility, especially during these huge events. So we know that these pulses are coming out now. We have a much better handle on when and how they're occurring. And the question is, how is it affecting the dominant corals at this reef? And we've really kind of focused on corals for a couple reasons. Number one, in general, they're good bioindicators for any given reef. They respond quite quickly to environmental stress. But more importantly, we know that in most systems that are benthic on the coast of Florida, the more coral that you have, the higher your fish biodiversity is and the higher your overall productivity is on a reef. And so it's a good way to track whether or not your system overall is doing well. Jeff's already introduced you to zooxanthellae. The other thing that all corals have are bacteria all over their surface. So the same way we're covered in bacteria all across our skin, our eyes, ears, nose, all on the inside of our guts, most of those bacteria are commensal. Most of them help us. And for corals, it's the same way. But just like us, corals have a few bacteria that are pathogens there lying in wait, waiting for you to get stressed out to start an infection. And corals have that same problem. So by tracking those changes in bacteria, we can also get a good idea of coral health. So we're looking at the individual responses of the coral, the responses of the zooxanthellae, and the responses of the bacteria all together to get an idea of what's going on. And we focused on those 2 coral species, the brain coral, Pseudodiploria clavosa, and the mountainous star coral, Montastria cavernosa. And we're looking at 3 different things. First, the gene expression of the corals themselves. Were they serving popcorn at 7, Dennis? They were. Anyone smell popcorn on the way in? The moment you smelled popcorn, your body turned on genes to start producing digestive enzymes. Signals all the time cause certain genes to turn on and off. And corals are the same way. So what we've done is design a system to specifically look at when stress genes are turned on to quantify the amount of stress those corals might be experiencing. We also look at the changes in the overall bacterial community on the surface of the corals, and then changes in both the abundance and composition of those algae, the zooxanthellae. So here's a look at those gene expression data. The way we do this analysis is using a technique called a microarray, but you can also do real-time PCR, or you can do RNA-seq, for those of you that are savvy with those kind of terms. But the overall idea is that we have copies of DNA Those copies of DNA are transcribed into a little piece of RNA, and that piece of RNA becomes the functional protein in our cell. So if we can grab those pieces of RNA as they're being made, we know which genes are getting turned on. So we set up a microarray that's only going to test to look for the kind of pieces of RNA that are made during times of stress. And it's going to tell us not just which kinds of RNA are there, but relatively how much of those different kinds there are. And so we've got about 148 different genes that we're looking at, and those genes are replicated across the entire array about 2,000 times. And what we found is that during discharge events, we get elevations of certain kinds of genes, those related to cell metabolism. In other words, they kick it into high gear and start to build back if they can. Tissue repair, not too surprising, and osmotic stress-related genes. So all three of these are ones we might expect to be elevated during times of stress, and indeed they are. The other thing we can look at is those shifts in bacterial community structure. And rather than show you a whole series of graphs that show you changes in bacterial community structure, I've tried to simplify it just a little bit. So each one of these dots represents an entire bacterial community on a coral. And if two dots are very close to one another, that means those two corals have very similar bacterial communities. If two dots are really far from one another, it means those two corals have very dissimilar bacterial communities. And we can see on this graph, the way they tend to clump out is by time. So we looked at differences among time and differences among site and found that when we collected corals mattered more than where we collected them from within St. Lucie Reef. Indeed, each season grouped out independently. So that means bacterial communities on corals are shifting over the year, which is not too surprising. But it also means that our sites are not significantly different from one another. And when we first started this study design, we kind of anticipated that the northern sites would be the most affected, and then followed by the central sites and the southern sites in this gradient away from the inlet. And we now know from looking at the salinity data, looking at the light data, that it's a big-time washing machine out there, and that all those masses of water get moved around and across the reef at one time. It's not that the northern site's any worse off than the southern site, per se. And so this also has implications in terms of how we might manage water quality on the reef. Rather than partition it into zones and look for gradients across those zones, maybe we should be treating this as one unit. The other interesting thing when we look at the coral bacteria is that if we compare the bacterial communities that we saw at St. Lucie Reef to samples that we've taken from Florida Keys, Virgin Islands, and the Bahamas, these corals are completely different. You'll notice that they're completely separated out, so very different, far away from everything else. We don't quite know why yet. One of our next steps is to do a little bit more sequencing work to figure out which unique bacteria may be driving these trends. But suffice to say, this is yet another indicator that shows that corals at St. Lucie Reef are fairly unique from those in the rest of the Caribbean. So lastly, we wanted to look at those zooxanthellae. And a quick and easy way to try to figure out whether or not zooxanthellae are shifting is just to count them, to look at the density of zooxanthellae or the amount of zooxanthellae per unit area of coral tissue. So we take our coral samples back to the lab. They look like this with the coral tissue all over them. We use a little water pick, the same thing you'd use to clean out your teeth, and blast all of the coral tissue off of that coral skeleton. So it's a bare coral skeleton like you see down here on the bottom. Then we filter the zooxanthellae out of that blasted tissue and count them. And then we divide that number by the total area of this little fragment. So we get a total number of zooxanthellae per square inch or per square centimeter, either one. What you'll notice here is a couple things. Number one, our two species are very different. So we have far more zooxanthellae per unit area in the brain coral than we do in the star coral. And that trend holds no matter when you measure. Again, we didn't see any significant differences among sites, so I've not even bothered to show you that. So this other idea that maybe site doesn't matter as much as we thought it did. But then we do see these subtle shifts over time, and if you do an analysis, this is significant. where June and February and June here are slightly lower than what is in August and October of this past year. So we see shifts in the total amount of zooxanthellae over time, and it's different for our two different coral species. But again, notice here, the brain coral is higher. The other way to look at this is rather than the number of cells, look at the amount of chlorophyll within each cell. And when we do that, things flip. Now, the star coral has more chlorophyll per unit cell than the brain coral. So that means although the brain coral may have more zooxanthellae, each of its zooxanthellae have less chlorophyll. And that may have implications for photosynthetic efficiency. Again, time matters, sight does not. And then if we put those two ideas together, so if we take into account the number of cells per unit area and the amount of chlorophyll per cell, we can figure out the amount of chlorophyll per unit area, right? Amount of chlorophyll per square centimeter. You'll notice it's kind of smushed these together a little bit, especially from June on. In other words, there's this kind of compensating mechanism. The diploria, the pseudodiploria, the brain coral, has more zooxanthellae, but less chlorophyll per zooxanthellae, so it kind of averages out. The Montastria, the star coral, has fewer zooxanthellae, but each one of those zooxanthellae has more chlorophyll, so it's compensating a bit. So that means a couple different things. Number one, we may have some kind of compensation effects such that they're both functioning somewhat similarly. But what we're also interested in is whether or not this is an indicator that these two corals house different kinds of zooxanthellae. There's at least 9 different zooxanthellae clades that have been described. Most of them are not well resolved down to the species level. But we know that the kind of zooxanthellae you have can dictate whether or not you can handle elevated temperatures or whether or not you can handle less light. So we're going to take this a bit farther and try to figure out which exact kind of zooxanthellae are present to determine whether or not this really has an impact on the coral's health and survival. So to summarize those kind of 3 different ways of looking at coral health, we know that there's really significant increases in the gene expression related to coral stress during the discharge events. We also know that the corals that we have at St. Lucie Reef have this really dynamic microbial assemblage over time, and that it's completely unique from things we've observed elsewhere in the Caribbean. And lastly, we know that both density and chlorophyll concentration vary among our 2 coral species, but in this inverse relationship, which may have implications for who's there. So our next steps, first we want to look for those different kinds of zooxanthellae. So one of my graduate students, Courtney Claypack, has been working on an assay to quantify the relative abundance of zooxanthellae types. So that's going to allow us to do a couple different things. Number one, see if these 2 coral species have different kinds of symbionts, and if that means one's tougher than the other. And it's also going to allow us to see if over time, individual corals are switching the kinds of zooxanthellae that they have, or the relative abundance of the kinds that they have, to compensate for discharge impacts. One of the big problems that we have with this study is that we're in Florida. Sometimes it's hot and wet, and then it gets hotter and wetter, right? But when it gets hotter and wetter, it happens at the same time. So it's very difficult for us in doing a field experiment to tease apart the effects of heat from the effects of the discharge events. And so the way we'd like to do that is to do an ex situ mesocosm, or big aquaria out of the water, where we independently manipulate water, use either discharge water or clean offshore water, and independently manipulate temperature, either do ambient temperatures or elevated high summer temperatures, and look at the responses of the corals to see how they're reacting to these independent and combined stressors, to tease those 2 apart. And what we're really looking for here is not just the smoking gun, but we're worried about this idea of synergistic impact. So there's a probability that as it gets warmer, corals are less capable of dealing with discharge water. In other words, that there's a synergistic effect of both of these stressors. And if we find that to be true, that becomes really important for figuring out when we want to release our water. If we know corals are more susceptible when it's hot, maybe we don't do water releases during the hottest few weeks of the year, right? We also are looking to figure out which of these coral genotypes are most resistant to discharge stress. And there's a couple different reasons behind that. Number one, if we can identify which coral genotypes are resistant, we can start to probe in and look at their genes to figure out which of those genes may be conferring resistance. And furthermore, as more and more emphasis gets placed on restoration, in general, there's this trend towards trying to select corals that you think are going to be hardy and perform well if you grow them up in a nursery and then do outplants. Now, there's a risk associated with that. You don't want to favor a certain kind of genotype in the environment because it all gets back to biodiversity. If we select corals and their genes that are only good for the environment that we have right now, if that environment shifts again, we may have picked the wrong horse to bet all of our money on. So you have to do this with a note of caution. And then the last thing, and this is something that's just come up in the past few weeks, is I've been working with some colleagues at Smithsonian on developing a metagenomic eDNA approach. In other words, rather than going out and just looking at the responses of corals, grabbing whole buckets of water or whole buckets of sediment and looking at all of the DNA that's present in those buckets of water and sediment and looking for elevations or drops of certain individuals in response to these discharge events. And the idea behind this is that, ultimately, we know that the corals have implications higher up, the parts of the food chain that we tend to care about. So do all of these other small eukaryotic organisms. So if we can track shifts in the kinds of organisms that we know are food for fish, we can predict or even maybe forestall losses of fish that fishermen and both recreational and commercial fishermen care about. So we're really looking to combine the field monitoring that we've been doing with these controlled experiments with different kinds of water and temperature and developing these new molecular diagnostics to put them all together to create better data for both effective and adaptive management. Kind of the holy grail here is to get to the point where we've developed diagnostics that are quick enough that when a management decision is made on the order of months to a year or maybe two, we have a performance metric of whether or not that management decision positively impacted this offshore coral reef. Right now, almost all the performance metrics are focused inside the estuary itself. We're trying to take that next step so that we know what happens to that water once it goes beyond the inlet. And what would be ideal is we get to a point where we have positive data that shows instituting one of these performance measures has been successful. If we do that, it's going to help build momentum to taking additional steps towards restoration. And I think that's really the biggest hump that we have to get over from a political and will standpoint is taking on an expensive project and trying to determine whether or not it worked. And when it comes to the offshore coral reef, we don't have enough metrics yet to figure that out. So that's the gap we're trying to fill. There's a ton of folks to thank who have worked on this project, a host of students and interns and volunteers, et cetera, as well as funding from the FWC State Wildlife Grants Program, a donation from the River Branch Foundation, support from the Save Our Seas license plate, and really great cooperation from the Florida Park Service, who's been continually helpful in allowing us to access this location. So with that, I'll say thank you all for coming, and Jeff and I will be happy to entertain questions.